Neuroscience clinical trials are very expensive, and more than 80% compounds fail in mid or late stages of development.
How do you deal with regulatory compliance?
Your model is only as good as your data. You need to ensure that those additional data sources are also accurate. Machine learning models must be rigorously validated.
How do you ensure reproducibility, and also how do you address the bias? This is where we bridge the gap between innovation and scientific discovery. This is where technology meets science, a podcast by Epsilon. Each episode brings you insights from industry leaders who are pushing the boundaries of what's possible in life sciences. Enjoy, and if you like the series, click that subscribe button. This series is brought to you by Epsilon, a technology company that empowers pharmaceutical and life science companies to leverage open source technology for faster, data-driven decision-making in regulated environments. Welcome back to Where Technology Meets Science. This is Nat, your host, and today we have a guest from Jazz Pharmaceuticals, Jing Dai. Welcome. It's great to have you here.
Thank you, Nat. Nice to be here.
Jing is a director of Biostatistics at Jazz Pharmaceuticals. She has been a neuroscience and clinical biostatistician since 2017, and she has been working in the medical affairs and real-world evidence space since last year. Prior to joining pharma, Jing was an assistant professor and biostatistician at Wayne State University School of Medicine. Jing, I know you have recently attended a PHUSE US Connect conference in Orlando, Florida, and you have also presented there. Could you tell me more about your experience and what was your presentation about?
Thanks, Nat, for that question. Just a quick disclaimer here before we start that the views expressed here are all mine and do not represent any entities I'm associated with. For your question, PHUSE is always a great conference and a great experience. I learned so much about use cases of real-world data, data-based risk management, and the ever-evolving data standards. And this year, in particular, many people, including myself, have witnessed the impressive strides our industry has made towards the adoption of AI and machine learning for clinical trial curation and acceleration. There are great discussions on leveraging the latest large language models for TFL and CSR automations. There are demos of using powerful GPTs such as data analyst from the enterprise version ChatGPT to analyze real-world data and integrating ChatGPT into R and Python programming languages for real-time interaction and debugging. As for my presentation, I spoke about my experience helping my team streamline machine learning modeling in neuroscience clinical trials through multifunctional collaboration. In a nutshell, it's about bringing a highly diverse team together to leverage machine learning techniques to try to address a couple of challenges in neuroscience clinical trials for better trial designs.
Thank you so much for this. I have a little bit of FOMO missing the conference, but let's take a step back. And why do we focus on neuroscience clinical trials here? What are some of the issues that could be addressed with machine learning?
Great questions, Nat. Ever since I joined industry, I've been working with neuroscience clinical trials in the areas of CNS, psychiatry, neurology, and sleep. Neuroscience clinical trials are very expensive, and more than 80% compounds fail in mid or late stages of development. Despite all of the amazing discoveries that we have heard that's made in neuroscience in the past 3 decades, there are still quite a few persistent challenges, I would say, that our field are still faced with today. For example, our understanding of the underlying biology of the disease is still quite limited. Oftentimes, we don't really have clinically meaningful endpoints or validated biomarkers for the indication we want to study. At the same time, the patient populations can be really heterogeneous. And on top of all this, Many neuroscience therapeutic areas are also challenged with high placebo response and high attrition rate. Let me give you a couple of examples here now. In major depressive disorder trials, placebo response rates can be as high as 50%. In Alzheimer's disease trials, dropout rates have been reported as high as 40%. So, as we know, high placebo response can obscure your true treatment efficacy, leading to false negative results. And on the other hand, high attrition rates can result in substantial missing data, rendering your trial results less generalizable and less interpretable. Together, these two issues can really drive up costs, delay the drug development timelines, and contribute to the high failure rate of neuroscience compounds. And my work actually focused on how to effectively leverage machine learning in interdisciplinary setting to help address the high placebo response and high attrition challenges. We are zoning in on these two specific issues here because when the trial fails or gives mixed results, these are usually the most recognizable and diagnosable factors that stand out in the data compared to other, let's say, bigger challenges like disease biology or endpoint selection. Placebo effects and missing data due to attrition are actually straightforward, or say more straightforward, to quantify and model. And by tackling these challenges, we can make a tangible and immediate impact on trial design and data interpretation in the current drug development cycle.
It's very interesting, and it's also there's a lot of amazing work you've been doing there. But let's dive deeper into machine learning. So, how can this machine learning models identify patient subgroups or biomarkers predictive of high placebo response or dropouts in neuroscience trials? Why didn't you use traditional statistical modeling?
Yeah, and those are great questions, and we can go really technical on these, right? But to sort of like briefly answer the question, we really need to take a look at the modeling objectives here. In neuroscience clinical trials, oftentimes we find ourselves at a phase two readout, and the pressure is on to quickly identify those who are likely to drop out and those who are likely to respond to placebo based on their baseline characteristics, because these insights could be critical for refining our phase three designs. The problem is that in phase two, we often work with very small cohorts, sometimes 100 patients or fewer. Yet, we typically collect a lot of baseline data, sometimes even more than what we would collect in a much larger phase three trial. This is because phase two trials are not just about detecting the efficacy signals, they also serve a critical purpose for hypothesis generation, which drives the need for deeper and more granular data collection. But Nat, this actually creates a typical small n and large p problem, right? N means your sample size, p means the predictors. So you have a small sample size and large number of predictors. So this is where the traditional statistical models like logistic regressions struggle with high dimensional and highly correlated data. Machine learning, on the other hand, actually excels in this setting. It can uncover complex, nonlinear relationships among patient characteristics, automatically select the most relevant features while tuning the model at the same time. And by leveraging these strengths, we can extract meaningful insights from these small cohorted but information rich data sets. Um, I know that's a short answer for a big question, but hopefully it sheds some light on it.
Thank you. That explains a lot. And how do you deal with regulatory compliance? What frameworks and guidelines do you follow to ensure that those machine learning models meet the regulatory compliance of different regulatory bodies?
Yeah, that's actually very important to discuss here, Nat, because we don't want to just go on the technical side without thinking about regulatory compliance. Because I would say that even though this is still a relatively new area we're navigating here, the fundamental principles actually are not that different from what we already know in statistical modeling, which is the field we're all more familiar with. That's why I would say the GXP framework remains the most important guidelines, especially now that FDA and a couple of other health authorities have recently come together and released the GMLP, or Good Machine Learning Practice, which outlined 10 very important guiding principles for machine learning in the medical device space. So these GXP guidelines have already provided a sturdy framework that ensures the integrity, quality, and traceability of data and processes. But when focusing on machine learning modeling with clinical trial data, I think there are a few particular areas that we could follow to ensure regulatory compliance. First and foremost is the data integrity. Now, your model is only as good as your data. And this does not change. If you're working with clinical trial data, you're likely starting from a very solid background because of the high level of rigor of most clinical trials. But if you're also looking at integrating data from external sources, such as academic collaborators, specialty vendors, or real world data sources, then documentation becomes absolutely critical because you need to ensure that those additional data sources are also accurate, reliable, and properly tracked. And next, of course, we need to ensure model validation. Under GXP, machine learning models must be rigorously validated to demonstrate that they perform as intended. This includes verifying the algorithm meets the performance criteria. One of the GMLP principles, for instance, actually spell out that the training and the test data sets that the model is evaluated on must be independent from each other. This ensures that the model is evaluated on data it has never seen during training or tuning, and the performance metrics are assessed on how the model will behave in the real world. And we also need to ensure regulatory readiness by fostering compliance and transparency, just similar to what we already do with statistical analyses and modeling. The software and computing environments that you use must be qualified and compliant with regulatory standards. If you can pre-specify your modeling objectives, methodology, and the performance criteria, you should absolutely do that. However, if you cannot pre-specify, it's not the end of the world, but it's best to engage with regulatory agencies as early as possible to get their timely feedback. You also want to make sure your machine learning results are reproducible and your decision-making process or the workflow is explainable. I cannot stress this more because this is crucial not only for regulatory acceptance, but also for future audits. And that I also want to add that in addition to the GXP and the GMLP that we just talked about, FDA actually also released a draft guidance earlier this year, specifically focusing on the use of AI to support regulatory decision making for drugs and biologics. It outlines a risk-based credibility assessment framework, which actually builds on what we just talked about. And this framework basically asks the researchers to take a few steps, right? So you need to define the specific question your model is addressing. You need to clearly articulate the context of use. You need to evaluate the risk of what if your model prediction is wrong. And finally, you need to create a credibility plan that covers all of the technical details in your model. And you also are encouraged to have a plan. Not, everyone is, applicable for every situation here. But if it's applicable for you, you are also encouraged to have a plan for ongoing monitoring and maintenance to make sure your model stays as reliable as conditions change and evolve as you intended for the original conditions that the model were trained and built on. So there's so much more detail that we could actually dive into. But hopefully that covers your question, Nat.
Thank you so much. But you mentioned reproducibility earlier, which is a very important element of all of different elements of our computing environment. So I just want to ask you, how do you ensure reproducibility within your machine learning modeling work, and also like how do you address the bias?
Yeah, these are great and important questions again, Nat. And I would actually like to start with reproducibility, um, as it's more of a technical challenge, right? But bias really can be very complex because it's multifactorial, it could be multi variant. So sometimes bias is much harder to assess and to address. So the good news is that our field of machine learning has matured enough for us to have the tools that we need to ensure the reproducibility. Today, we have tools and platforms like Git, DVC, and machine learning flow that support data and model versioning and experiment tracking. And in both R and Python environments, you can actually use version controlled scripts, and you can make sure the markdown files are maintaining a reproducible workflow. And other basic things to cover here, for instance, for every step of your model, no matter it is data splitting, imputation, bootstrapping, oversampling, or the model fitting itself, you can always use a consistent random seed to ensure results can be reproduced. Now, coming back to bias, I think the most critical part here is actually to identify sources of bias early on. For example, is the bias due to underrepresented demographic groups? Is it introduced during feature selection? Or does the bias actually come from the modeling strategy itself, such as possible data leaking in longitudinal modeling? So once you have pinpoint the major sources of bias, there are usually established methods for mitigating the bias. For example, if a key demographic group is underrepresented in your data, a number of oversampling techniques can be used to generate synthetic samples to boost the representation so that the model does not disproportionately favor the dominant group. If your features were handpicked by domain experts, such as clinicians that I work with, you can always involve a more diverse group of expert panel to bring in different perspectives and reach consensus on what are the really clinically relevant features to select. You can also validate these features using data-driven methods like recursive feature elimination to check that they actually hold up statistically. And for the bias caused by future data leaking into the training set, in longitudinal modeling, you can use time-based data splits or rolling window validation methods to maintain proper separation between the training and the test sets. But Nat, here is the tricky part. It is really hard to make sure that we are not introducing new bias while addressing the existing ones. Yeah. So multiple imputation, oversampling, binary classification, even just creating composite variables can all produce an extra layer of bias. In fact, the more you engineer your data, the more bias you could potentially introduce. So oftentimes, yeah, you will find yourself performing sensitivity analysis with your machine learning models, just as you would do with your traditional statistical modeling, especially in clinical or real world evidence setting, where assumptions can heavily influence conclusions. And this is human safety we're talking about here, right? So you need to pressure test your model to make sure the model is valid and robust under alternative assumptions. So that's why I said in the beginning that compared to bias, reproducibility is actually not that hard to address.
That sounds like a lot of steps where a lot of things can go wrong. So, do you know any examples, precedents where the insights from AI and machine learning modeling have been accepted by the regulatory bodies and successfully integrated into the future trial designs?
Absolutely not. There are actually very encouraging examples and great precedents to show how far our field has come. So I would say ever since 2016, there have been dozens of FDA approved AI and machine learning based medical devices and algorithms, with radiology and cardiology leading the way. Many of these tools function as digital biomarkers by converting digital data into objective and quantifiable indicators for disease diagnosis. One notable example is the IDXDR system. And this is one of the first, if not the first, fully autonomous AI diagnostic tool cleared by FDA to detect certain diabetes related eye problems using digital imaging. It sets a precedent for how AI developed digital biomarkers can be integrated into clinical workflows, offering patients a non invasive, reliable, and objective method for early disease detection without the need for specialist interpretation. And because we're talking about drug development, I also want to share another example that's closer to home, where machine learning not only received regulatory buy in, but also shaped the design of a future clinical trial. I'm getting very excited talking about this net. So, this is actually a study published just last year. Researchers introduced an enrichment design approach to make bipolar depression trials more sensitive. Using data from previous trials, they actually applied a machine learning model called isolation forest to identify patients whose symptoms followed a typical and well structured symptom pattern. And they also flagged those patients with atypical or unusual symptom patterns. And they signed each patient with a symptom anomaly score. Then, in the retrospective analysis of a phase 2 trial, they found that patients with low anomaly scores demonstrated significantly higher effect size with better signal detection and reduced placebo effect. So, encouraged by these results, the enrichment algorithm was prospectively implemented in a phase 3 pivotal trial, making it one of the first examples of subject level machine learning based enrichment applied in real world regulatory contexts. Just to also mention that this approach is absolutely consistent with FDA's guidance on enrichment strategies for clinical trials. So the guidance, in fact, encourages the use of patient characteristics to improve trial sensitivity. So in this study, by reducing symptom heterogeneity, their strategy helped increase trial efficiency and statistical power without increasing the patient's burden. And so this is a great win net, right? Because particularly in psychiatric trials, we have subjective measures, we have high placebo response, and these are major challenges. And this study really helped set a precedent for that.
Very interesting. Thank you so much for this, Jing. We are slowly getting to the end of our conversation, but I just want to also refer to your PHUSE talk, because it's been through all of the conversation on the podcast and not on the podcast, I've been hearing that the talent and the fact that there's so many people working together and pushing to move to open source. So basically, the talent is just like one of the most crucial elements of having innovation, pushing the boundaries of clinical trial design. I want to ask you, because you spoke about the interdisciplinary collaboration. So, what, in your opinion, are the biggest challenges you have encountered in machine learning? And there are multifunctional settings. And what steps do you take to foster collaboration between different skill sets, different people like biostatisticians, clinicians, and engineers?
I don't know if my answer is going to surprise you, but the biggest challenge I have encountered is actually the communication part, right? So it's about getting everyone together, including like the roles and functions you talked about, biostatisticians like myself, statistical programmers, data scientists, and clinicians, and also other functions like regulatory folks on the same page, especially under very aggressive timelines, because most of this work, right, at least in my experience, happens at end of phase 2, where we're interpreting results, running postdoc analyses, designing phase 3 studies, and maybe also preparing a briefing book for regulatory discussions all at the same time. And each function group has their own language, their own perspective, their own way of thinking. So it's very important to align well and align early. Otherwise, it's really easy to run into confusion later on about modeling objectives, data issues, or simply what the results actually mean. Process-wise, I've found it really helpful to establish a structured and repeatable workflow. So the workflow usually starts with a cross-functional kickoff meeting where we align on modeling objectives, timelines, resources, roles, and responsibilities. Once these expectations are set, the work naturally flows into data integration, feature engineering, followed by the actual machine learning modeling phase. Another critical piece, I believe, is to make sure the non-modelers on the team can actually understand and confidently act on the machine learning results. That means putting in extra effort to simplify technical concepts and foster knowledge sharing. For example, rather than diving into complex math, we rely on intuitive diagrams and animations to explain how the models work at a higher level. We also create practical tools, and don't laugh at this, such as cheat sheets that would tie machine learning terminologies back to the more familiar statistical concepts, such as when we'll have a table, the left column would be machine learning, the right column would be statistical concepts. And then, so we can see that when we say features, we mean independent variables. When we say recall, we mean sensitivity. When we say loss, we mean prediction error. So this way we can demystify machine learning and help our non-modelers get comfortable with the new terminologies, and in turn make team communication much more efficient and transparent. And one last thing I want to share is that we actually keep everything well documented, right? Not just for regulatory purposes, but also for internal communication. And we meet frequently. So it's easy for the whole team to refer back and stay aligned. And at these regular meetings, we revisit project progress and we adjust the modeling objectives if necessary. And we also do knowledge refreshers to reinforce the understanding. So that's how we address the biggest challenge of functional alignment.
Yeah, that's very interesting, especially the cheat sheets and translation tables.
Yeah.
I think a lot of our listeners can use it as an example, because the using different language different languages between different disciplines is probably one of the biggest challenges, and not only in the pharmaceutical industry. Okay, the final questions that we ask to all of our guests. What is a problem in your field that you are surprised that has not been solved yet?
This is my personal opinion. I am honestly surprised that in 2025, we're still relying on highly subjective endpoints in psychiatry and many areas of neuroscience. Diagnosis and outcomes often depend on rating scales and patient-reported outcomes that can vary depending on who is administering them, the mood of the patient that day, or even how the questions are asked and phrased. And these scales, this is another probably bigger problem, is that these scales are often very lengthy and time-consuming to administer. Some would take an hour and a half, 2 hours to complete, which creates a huge problem because they are rarely completed in full during routine clinical care in the real world. This creates a clear gap between how data is collected in clinical trials versus how data are collected in real-world settings, making it very hard to generalize the results or align the regulatory and real-world evidence. We really need better assessment tools and more reliable biomarkers. Of course, all of these will be based upon a deeper understanding of the diseases themselves, right? But looking at the brighter side, Matt, I do believe that AI and machine learning will play a key role in moving us forward. And we're starting to see a lot of promising signs. For example, the QB check is a FDA-approved AI-enabled tool that supports ADHD diagnosis by objectively measuring core symptoms through motion tracking and task performance. It helps reduce subjectivity and it brings consistency to clinical assessments. And in depressive disorders, we see deep learning models being developed to supplement or confirm the clinical diagnosis through facial expression analysis. And in Parkinson's disease area, we see the development of highly accurate AI-based digital biomarkers, which can analyze breathing patterns during sleep to diagnose and monitor the disease, even predicting the risk years in advance. So I would say these are great steps forward, but we need more of them for sure. So my hope is that in the near future, psychiatric and neurological assessments will become much more precise and data-driven, ultimately helping us design better treatment and clinical trials and improve patient outcomes in a more efficient manner.
Thank you. I think we can end on this positive note. Improving the patient's life and their health. This is our purpose. This is what we aim for. And then all these amazing tools that we talked about today are hopefully gonna make this better, more efficient, and faster. Thank you so much, Jing. To our listeners, if you feel that you know somebody who would find this episode interesting, you can share with them. And I encourage everyone to subscribe to the podcast so you're not gonna miss another episode. Again, thank you, Jing, so much. It was very insightful, a very nice conversation. It was lovely having you here.
Thank you, Matt.
Thank you for listening. This was Where Technology Meets Science by Epsilon. If you know someone who might enjoy the episode, share it with them and subscribe to never miss a new episode. At Epsilon, we empower pharmaceutical and life science companies to leverage open source technology for faster data-driven decision-making in regulated environments. Until the next time.